So we are doing biology test 1 for 2021. So we'll project the questions, the first question for that paper. Okay. So we have question number 1 there, which is saying briefly describe how you would use a right microscope to view a slide. Okay. So we are told to describe how we use a light microscope to view our slide there. This was 5 marks. Okay. So how would you use a light microscope to view your slide? What are the steps you need to take? So this is the first topic, and these questions came. This was 2021 test 1, and those are the questions which came. So how would you use your light microscope to view a slide? You have 5 marks there. What are some of the steps you would follow? Okay. Okay. So the first step you want to do, okay, you would want to position your specimen or your slide, okay, so that it is over the source of light. And remember, when we are doing the first topic, we learned that the source of light for a light microscope is going to be an what we call an illuminator, isn't it? Okay. So the first thing, you would place your slide. You place your specimen on the slide. Then you get your slide. You put it on stage. Okay. So you place your slide on stage. Okay. Then on that stage, you have the specimen on that slide. Okay. Then you place it on stage so that it is over the source of light, which is which we refer it to be an illuminator. Okay. So that's the first step you would take when viewing a microscope. So you give points like that. So you get a 1 mark, a 1 point there. Okay. Then what is the next step after you place the slide on the stage? What step are you going to take next? Okay. So what step would you take after putting your slide on stage? What's next now? Those who've been studying, I don't know if you haven't been studying. It's high time you started studying, because time moves. Very soon you have your test 1. So what do you do after placing your specimen on slide? Then you put it on stage. What's the next thing? Okay. So you start viewing your slide, isn't it? Your specimen. Okay. So you start with the low power. You start. So you view your specimen starting with the low power, or starting at the low power. Okay. So it can be low power. You start with times 4. We are talking about the magnification here. So you can start at times 4. You can start at times 10. Okay. So this is where you view your specimen at low power. That's the next step after putting it on stage. Okay. So you start viewing it. You start with the low power there. Okay. Are we together up to here? Okay. So you have your second point there. Okay. So you put your specimen on a slide, then you put it on stage. You view it starting with the low power there. That's the next thing you do. Okay. So we have 2 points there. What's the first step you take when viewing your slide using a light microscope? Apart from these two mentioned, what's the next step? So the first step, we said we place our slide on the on stage so that it is over the source of light, okay, which is an illuminator. Then the second step, we said we start viewing our microscope. We start with the low power. So you start with the low power. So as you are moving, you'll be increasing. So you start at low power. You can start at times 4. in terms of magnification. You can also start at times 10. Those are low powers. Okay. So that's the next step. That's the second step you do. What's the first step? Okay. So what's the third step? Those who've been studying. What's next? Low. Okay. Okay. So what's the next step after you you view your slide at low power? What do you do next? Okay. So you have to focus your image, isn't it? That's the next thing. Okay. So you focus your specimen. Okay. So what do you use to focus your specimen? What do you use? What part of the microscope you are going to use to focus your specimen? Hello? Okay. So you use what we call a close focus knob. Okay. So you use your close focus knob to focus the image. Okay. So that's the next step. So you place your specimen on a slide, you put it on stage, you view it at low power, then you focus it using your close focus knob there. That's the third step. What's the fourth step? Okay. So you have three points up to here. Okay. So what would be the fourth one? What would be the fourth step after you focus your specimen? What would be next? Okay. So you adjust the diaphragm. Okay. So you are going to adjust your diaphragm and light, isn't it? Okay. So you adjust or you regulate the amount of light using the what? The diaphragm. Okay. So you want to get a clear image here. Okay. So you adjust the what? The diaphragm here. Okay. You adjust the diaphragm. Okay. And the light so that you get a clear image. Okay. That's the first step. Okay. Then what can be the final step? What would be the final step? What would be the final step? Okay. So now remember at point number 2, we were viewing our slide at low power. Okay. So the next thing after you get your clear image. Okay. So here we have adjust. Then we are getting a clear image. Now we can view our slide at high power. Okay. So that would be the last step. Okay. The final step, you switch to what? So you switch that magnification to high power. So you switch the magnification to high power. Okay, so these are some of the steps you are going to take to when viewing your slide using a light microscope. Okay, so that was part A. So you get your 5 marks there. Okay, so let's go to part B there. Okay, I hope people will be participating so that the more you participate, the more you get to know what you've been studying is correct or not. Okay. So these are this topic you've done in class, and I should be able to answer such questions. Okay. So now let's compare the advantages and disadvantages of a light microscope and an electron microscope. Okay. So let's get the advantages. Okay. So we start with the advantages. Since we have 4 marks, we're going to get 2 advantages. Then we are going to get 2 disadvantages. Okay. So let's get advantages. Okay. Of your we start with okay, so here I'll put light microscope. Okay, so get 2 advantages of a light microscope, and also we get two disadvantages. Okay, so let me do this. Okay, so I have advantages this side. So I'll get two advantages and two disadvantages for a light microscope. Then I also get two disadvantages and two advantages of an electron microscope. Let's start with the advantages of a light microscope. Okay. So what are some of the advantages of a light microscope? Anyone who can try? Anyone to give it a try here? Okay. Any advantage of a light microscope? Let's start with a light microscope. What are the advantages of using a light microscope? Okay. So remember when we had that lesson, okay? So remember we said an electron microscope, we only use it to view things that are not living, okay? So we cannot use an electron microscope to view things that are living. Okay. So the advantage of this light microscope is that it can view even living things. Okay. So you can use it to view both non-living things and living things. Okay. So you can use it to view living spacemen. Okay, so let's use spacemen there. Okay, so that's one advantage of a light microscope here. Okay, okay. What's the other advantage of this light microscope? Okay, so we have one there. Let's have our point number two. Okay, let's just get two advantages of a light microscope. Okay, so you can also say that if we compare this light microscope, okay, so it is very easy to use compared to an electron microscope, okay. So we can say it is easy to use compared to an electron microscope. That's another advantage of a light microscope, okay. So it is easy to use. It is also cheaper, okay. So it's cheaper than an electron microscope. That's another advantage. Okay, so we've given about three advantages here. So those are enough, okay. Now, what are some of the disadvantages for this light microscope? Remember, we've started with light microscope. What are some of the disadvantages? Anyone? Okay. So one of the disadvantages for your light microscope is that it has lower resolution. Okay. And remember, we related resolution to how visible things are. We said the higher the resolution, the better the image, isn't it? So this light microscope has lower resolution. That's one of the disadvantages for a light microscope. Okay, so it has lower resolution, and we are saying the greater the resolution, the better the image. Okay, so the greater the resolution, the better. Okay, so the better the image, and we are saying this light microscope has a lower resolution. Okay, that's one of the disadvantages of this light microscope. Okay, also the magnification is also low. Okay, so it has lower magnification. Okay, okay. So that's another disadvantage of a light microscope. Okay. So now let's go to an electron microscope. Okay. So let's go to an electron microscope. So people should be able to tell me disadvantages after I mentioned the first point on the advantage of a light microscope. Okay. But let's start with the advantages of an electron microscope. Okay. So what are some of the advantages of electron microscope? Okay. So here we said this light microscope, it has low resolution, isn't it? So he said it has lower resolution, but this electron microscope, it has high resolution. Okay, so it has high resolution. That's one of the advantage of this electron microscope compared to a light microscope. Okay, then even the magnification is greater. Okay, so it has greater or high magnification. Okay, compared to a light microscope. So it has higher magnification. Okay. So those are some of the advantages of your electron microscope. Okay. So let's go to the disadvantages. Anyone to tell me one of the disadvantages? Anyone? If you've been following, what can be a disadvantage of this electron microscope? Let's get even two. Okay. What can be yes?
It is expensive
It is
compared
expensive.
to
Yes, true. So it is very expensive compared to a light microscope. Okay. What else? What else apart from that? Okay. So remember, I said this light microscope, one of the advantages is that it can be used to view living specimen, isn't it? But for an electron microscope, you only use it to view dead organism or specimen. You only use it to view non-living specimen. Okay, so that's another disadvantage for this electron microscope. Okay, so you cannot use it to view a living specimen, isn't it? That's another disadvantage of this electron microscope. Okay, so you get your full 4 marks there. Okay, so is everything clear up to here? Are people okay up to here? Okay, so immediate when you pass through questions like this, what you do is to go back, you check your materials. The materials you've been studying itself is not enough. So you need to be going through questions like this one, not because they will repeat, but they will guide you on how you are supposed to be studying. Okay, and they will tell you how questions come. Okay, so you can study and then you think that you know it all, but you find that questions are different. So these questions, the more you go through, the more you tend to get familiar on how these questions are asked. Okay, so normalize passing through questions like this. Okay, so you can go to part C there. So what does it mean when a microscope has a resolution of 0.2 uh, nanometer? So the resolution is very small there, 0.2 nanometer. You have 2 marks. What do you think it means when a microscope has a low resolution? Okay. So you guys, you did this in class, and we also did when we did microscopy. We talked about resolution. So what does it mean when uh, a microscope has this resolution? So it has this small resolution, which is 0.2 nanometer. Okay. So what does it mean? How can you get your 2 marks here? Okay. Anyone to give it a try before I... Okay. So so if a microscope has a resolution of this one or this fine, this is a very fine resolution. Okay. So it means that this microscope can only identify structures which are very fine. Okay, so it can be it means that the microscope, since it has low resolution, which is this one, 0.2 nanometer is a very small resolution. Okay, so means that it can only be able to show, okay, very fine structures, okay, structures, okay, which are closer to that resolution, isn't it? So which are closer uh, uh, to 0.2 nanometer. Okay, so that's what you have. So that's what it means here. Okay, so it means that this microscope, it can be used, it can show very fine structures there, which are closer to that diameter, which is a 0.2 nanometer there. Okay, so that's what you have here. Okay, then you have part D there, which is saying briefly explain how the invasion of the image uh, or the microscope. Okay, so briefly explain how the invasion of the microscope is important to life science. Okay, so you have 6 marks there. So what's the importance of invasion of the microscope in life science? So now let's apply. So you know what invasion is, and you know what invasion of the microscope is. Okay, so why is it important to life science? Okay, so you have 6 marks. Okay, so what do you think the invasion of microscope? Okay, so what's the invasion? Or we can use simple terms. The introducing, isn't it? Let's say the introduction of a microscope. Okay, so what do you think it's important to life science? What do you think it's important? Okay, so what's the importance of microscope in short? Okay, so this is the first topic. This is something you learned first in class. So you should be able to give points here. So what's the importance of microscope? Anyone? Okay. Anyone to give us some of the importance of this of microscope in science? Okay. So when we are doing the cell theory, okay, you notice that some of the guys started discovering cells. Okay, and the first guy who discovers the structure of the cell, I remember that guy. He got a piece of a part from a plant, then he viewed it using a microscope, isn't it? Okay. So in short, one of the point I'm trying to raise here, the importance of microscope in science, you can use it to for discovery. Okay. So it's the one which was used to discover cells. Okay. So you can say used in discovery of cells. Okay, that's one of the importance of this microscope. Okay. What's the other importance of microscopy? So we have one. We have to give about 6 importance of microscope. Apart from discovering of cells, what else? What's the use of microscope in science? Okay, and we know that these cells are very tiny. We cannot see them using our naked eye. Okay, so we cannot see the parts of the cells using our naked eyes, isn't it? So for us to understand those structures of the cell, the structures of these same cells, we have to use a microscope, isn't it? So we can use it to understand the structures of a cell and their functions. That's another importance of microscope in science. Any other importance? So we have two here, we've given two. Any other importance of microscope in science? Okay. So for us, sometimes, okay, so if you go to the hospital, okay, so you can use, they can get a part, let's say a biopsy, okay, so they get a biopsy, then they will use it, maybe they will view on the microscope to diagnose the type of disease you're having, okay, so you can also use this microscope for diagnosis of diseases, isn't it? Diagnosis of diseases. Okay, that's another importance of microscope. Okay, so given about four. Okay, let's give two more. Okay, so we can use microscope for research, isn't it? We can use it for research. Okay, also we can say we can use it in research and in biotechnology, isn't it? That's another importance of this microscope. Okay. What else apart from that? So we have four. Let's give one more point. We have five. I've given five. Okay. So the last one should be study of tiny organism. Okay. So study of tiny organism, which we call microorganism. Okay. So we cannot see those small organisms using our naked eyes. We need to use a microscope to view those tiny organisms. Okay. So those are some of the importance of a microscope in science. Okay. So let's go to part E there. Okay. So, okay, part E is saying with clear illustration, explain why different kinds of bacteria react different to gram negative, gram stain. Okay, so why do they react different to gram stain? Okay, so we did gram staining, and I'm sure you've also learned this in class by now. Okay, so why do you think different kinds of bacteria react different to gram stain? Okay, so we have two types of bacteria. We have what we call gram negative bacteria and gram positive bacteria, isn't it? Okay, these are the two types of bacteria we have. And what you need to know about these two types, you need to know their differences in terms of the structures. Okay, so you need to know what this gram positive is composed of and what this gram negative is composed of. Okay. Okay. So all bacteria have what we call uh, they have a cell. So all bacteria have a cell. So in this cell, embedded in this cell, uh, there's a carbohydrate, okay, called peptidoglycan. Okay, so this is a carbohydrate embedded in a cell of a bacteria, peptidoglycan. Okay, so now depending on the amount of this peptidoglycan in the cell of the bacteria, you find that this bacteria will be able to react different to this gram stain. Okay, so you have some bacteria with the thick peptidoglycan. Then we have some bacteria with the thin peptidoglycan. Okay, so for example, gram negative bacteria, this one they are going to have thin peptidoglycans in their cell. Okay, so they have thin peptidoglycan in their cell. Okay, and then gram positive, they will have thick peptidoglycan in their cell. So they have thick peptidoglycans. Okay, so that's one of the difference you need to take note of. Okay, so now in addition to a peptidoglycan, in this gram positive bacteria, you won't find what we call lipopolysaccharides. Okay, so there will be no lipopolysaccharide in a gram positive bacteria. Now, in a gram negative bacteria, you find what we call lipopolysaccharide. Okay, so these are the ones which normally contribute to gram staining. Okay, so for example, a gram-positive bacteria will retain what we call a crystal violet. Okay, so if you use a crystal violet stain to a gram-positive bacteria, it's going to retain that color, the crystal violet, because of the thick peptidoglycan. Okay, so it will retain the crystal violet color. Okay, now this one, the gram negative, has a thin peptidoglycan. Okay, so because of this thin peptidoglycan, you find that this gram negative bacteria won't be able to retain the crystal violet color. So with the reaction to the gram stain, you find that this gram positive end up having that purple color or violet color, which will be retained because of the thick peptidoglycan. Then here we end up having that orange color. Okay. Okay. So that is with the crystal violet. Okay. So that's a good example of why these bacteria react different to a gram stain. Okay, because of the composition of their cells. Okay, so I've mentioned two important composition of the bacterial cell, which you need to know. So you won't get marks without mentioning the thick peptidoglycan and the thin peptidoglycan, and also the issues to do with lipopolysaccharides and where you are going to find the polysaccharides. With these two, you even give an example like the way I've given here for crystal violet, should be able to get full marks there. Okay, so that's a reason why you have these bacteria reacting are different to your gram stain. Okay, so I hope everything is clear there. Okay, I hope all is well, unless there's a question. Okay, so we can do the last one there, part F. There is no question. We can do part F, which is saying give an explanation why basic dye are more commonly used than acidic dye in staining. Okay, so why do you think we use basic dye than acidic dye in staining? Why do you think we do that? Okay, so why do you think we use a basic dye than acidic dyes? Okay, so why do we use basic dyes than acidic dyes in staining? Okay. Mm. So if you look at the cell of most of these structures, okay, so if you check the organelles, and the organelles, we are talking about the cytoplasm, okay. Okay, so if you take you check the nucleic acids of these molecules in this structure, okay. If you check the cell walls, okay, you find that in these cellular structures, okay, so these cellular structures are okay. So these cellular structures are actually negatively charged, okay. So these cellular structures, most of the cellular structures are negatively charged, okay. So you find that uh, these are the basic dye, okay. So they usually react to negatively charged structures, okay. So you find that most of the structures for most cells, okay, you find that they are carry a negative charge, and you find that most okay, so the basic dyes, they will usually react to negative charges, okay. Then acidic dye, they actually stain positive charges, okay. So because of this reason, because of these structures carrying negative charges, you find that most of the common dyes are going to be basic, because they are the ones which are going to actually stain most of these structures, okay. So it's as simple as that, okay. So that's the main reason why you're going to find uh, most of the stains are going to be basic than acidic. Okay, because you're having structures which are most of the structures are negatively charged, and they are going to be stained by your basic dyes there. Okay, so I hope that is also clear. It's as simple as such. Okay, so that was your question one for test one 2021. It only had two questions. The other question, we haven't done some of the parts. So when we finish covered in my micromolecules, I'm also going to solve the other one. Okay. So for now, we can end here unless there's a question. Okay. So we'll continue with our lessons on Monday. So make sure you register, and I'll be solving most of these past papers. I'll be solving them, then I'll be uploading on the website. So if you've subscribed, you should be able to find most of the solutions for your test. You use them to devise as you are studying. You can be using them for the vision, so that you just become familiar of how questions are going to be asked. Okay. So we'll end here unless there's a question. Okay. 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 So if there's no question, we end here. Have a best night.